Hello, good afternoon, all of you. Hope you can see me. I don't know whether I'm visible there,
Yes, you are
but
visible.
uh, we have delayed a little bit because of the internet problem. Today, my topic must be levels of organization, but it is fundamental of taxonomy. That is how I felt more appropriate. So please note my today's topic is fundamentals of taxonomy. I am from B.N. Bandodkar College, Dr. Sudesh Rathod, head of Department of Zoology. Uh, Bandodkar College is situated at Thane, and mm, I am also um, associated with your college, Dombivli and Dalvi College, and also was exam chairman last year. This year I have quitted. I welcome you all students. I don't know whether today there are lots of problem on the net. So network is not working good, but still I feel that you're hearing me. I'll be talking on fundamentals of taxonomy. Next slide, please. Uh, this is our syllabus, and in our syllabus we have levels of organization, symmetry, coelom, and metamerism under fundamentals of taxonomy. Thank you. Next. Levels of organization, if we study, it is detailed as a cellular and or unicellularity level. What is acellular and unicellularity? We are going to discuss under that. Then colonization of cells and cellular level and or multicellular level organization. Then tissue level, organ level, and organ system level organization. Next. Talking about acellular and unicellularity level, acellular means what? I would like to ask you one question. You must be knowing that what is the meaning of cell or battery. The figure is given here. On the top left hand side there, the small figure is there which shows dry cell. You might have studied the dry cell under chemistry lot many times. Why it is known as cell? Because it is a unit of generation of electric energy. Okay. On a right side of the cell, you will find the battery. People usually call cell as a battery, but it is not a battery. Battery means it is a arrangement of cells in different form, maybe serial arrangement or parallel arrangement. So right side diagram besides battery is known as beside the cell is known as battery. And this battery is producing a current which is essential for driving the circuit or driving the mechanism of the circuit. Okay, that energy is supplied together. All cells together work to make functioning the unit of that particular circuit. So this is how cell and battery mechanism is. Similar is the case in our body. What is there on left hand side you can see the onion peel cells. Onion peel cells are very simple cells. They are arranged in a particular hexagonal or polygonal cellular structure, and these cells are working together. Okay, and when they are working together, the onion grows, multiplies, and it survives. Okay, onion is not existing without these cells. All cells together form an onion, just like a circuit I have shown above. So onion is an organism, living organism, and it is plant. Okay. And this plant also have several small cells. Now, by now you must be confirmed about what is the meaning of cell. Cell is nothing but it is a compartment of particular body or organisms. Here on above circuit, green color circuit is there, which is nothing but a particular organization or organism. And this organism of circuit, it is not living, it is non-living organism or organization. So this all together work for particular system, and that's why it is functioning. Similar way, animal body also is made up of several small units known as cells, which are nothing but compartments. Now let me tell, first of all, I am just letting you know what is the meaning of cell. Okay, we are studying what is the meaning of cell. Usually biologists know that cell is a unit of body and which is made up of cytoplasm and nucleus, and it is embedded in cell membrane. This is how cell is. But why cell name is behind that? Cell name cell itself refers that it is nothing but division. Anything divided into compartment is called as cell. You might have heard about cellular jail. Cellular jail. Cellular jail. Jail means what? A jail which is comparted with small rooms, and this jail is filled with there the prisoners are kept. Usually, these prisoners are high conviction prisoners, and these prisoners stay in jail or that small room called as cell. Okay, separate from each other. So this is how cellular jail. Our body is cellular. What does it mean? That means exactly our body is comparted. It is divided. Okay. Why it is divided? Actually, we have seen in embryo, animal starts with single celled zygote and then it multiplies into separate compartment is nothing but cells mitotically after fertilization. Okay. So this mitotic division gives rise to variety of cells, and they then differentiate into different kinds of cell, maybe nerve cell, muscle cell, and so. So this is how human body or any higher multicellular animal have that body. That is how a cell is. But now look at amoeba. On left right hand side, we can see amoeba. Okay. This right hand side amoeba, that's right. The pointer is showing now. So this blue color amoeba is a unicellular organism and there is no compartment inside body, just like multicellular organism. But what is the magic behind it is that this animal exactly show all functions as multicellular organisms show. Okay. So we can't say that it is not organism. It is organism because what are the properties of living organisms? Property is that they can first of all metabolite. So amoeba shows all metabolism, catabolism, anabolism, both can be seen. Then they grow after feeding. They eat the energy, food energy, and convert into body part. So they grow. Growing is there possible. Then they multiply or reproduce. Reproduction is also property of living organism. And lastly, very important is irritability and movement. Irritability is what? Animal respond to different stimuli. If you put any acidic dilute acid medium around amoeba will start to run away from that because it is not harmless, it is harmful. And because of that, animal can sense that it is harmful, I should run away, escape from this environment. On the contrary, if you put sugar sap nearby amoeba will get attracted because the amoeba can sense that it is my food. I can go close to this particular environment. So this is how irritability is expressed. Also, another property, if we are talking about animals, then they have one more property is movement, which is not seen in plants. Plants can't move, but animal can move. So that is how movement is an additional property of animal living organism. So if I'm talking about amoeba, it shows all this. So they are just like multicellular organism, isn't it? But what I want to say here, this animal is not comparted like multicellular organism. So although these animals are known as these hello, thank you. There was little problem. Uh, Again, a little connection problem. I'm very sorry. I beg a pardon for this disconnectivity. This will happen today because there is little problem in the network itself. So I was talking about amoeba. It is acellular because it is not comparted, but it plays all the role which are exhibited by living organism. So it is a complete living organism, but not cellular. And that's why it is known as a cellular organism. Now, unicellular is different. This is a concept because it is not cellular. It is usually referred as unicellular because one-celled animal. But if I'm talking about organism, this is an organism which is not comparted like multicellular organism, and that's why it is referred as acellular. Now, talking about next part is that here animal shows multicellular evolution into multicellularity. I think one slide is skipped. Can we go to earlier slide, please? No, you are going next one. Earlier. Previous one. Previous. One more. One more, please. This is what we were discussing about acellularity. So amoeba is acellular. In older taxonomist concept, this animal is always referred as acellular. And recently, some scientists have started using as unicellular organisms because they are made up of single cell like. But if I talk about organism, then at organism level, this animal is not comparted like multicellular organisms. So that's why it is known as acellular. Next. Next slide. Okay. This is how here this diagram shows that single-celled animal tend to become multicellular in the course of evolution. On the top, you see the dinoflagellates. These are protozoans. They belong to Masticophora. Masticophora flagella-bearing animal. There are certain animals which are singular. And I would like to say, first of all, Chlamydomonas is single-celled. Usually, they come into 4 tetrad form, like gonium during reproduction. Okay. Gonium is tetrad form that remains permanent in their life. They become into colony, small colony of 4 called as tetrad. Then Pandorina shows more number of individual together forming colony. So this colonization ability, then we go to higher level, Yamagishila, then Eudorina, Pleudorina, and Volvox. Okay, these are forming more and more number of individual in a colony. when you go to volvox, you will find internally it is having also the daughter colony. Even colony itself is made up of thousands of individuals, and inside there are small colonies known as daughter colony. The major colony, bigger colony is called as mother colony. Mother colony gives so many number of daughter colony, and this animal together live happily. And it is seen that animal gets lots of benefits by forming a colony in colonial form as compared to individual. So in individual, they are exposed to enemy more as compared to colony. Their chances of getting food is more as compared to single individual. Again, colony can locomote in a motion. These volvos can beat their flagella in a coordinated manner, and this colony can roll on surface of water very easily from one place to another. So that is how they become more efficient, protected, and assured for survival. And because of that, colonization was first attempt in these protozoans, who first of all thought or maybe tended towards becoming multicellular. And this is how this colony shows. Now, if you compare this colony just above part, green part, that is shown all dinoflagellates, those showing higher and higher grade of colonization. Similar is the case about our embryo formation. What is there? It is seen single cell starts dividing into two, and then four, then eight, and so on. It goes on increasing in number by doubling the number of the cell. And this morula stage is nothing but it shows in a colony of volvox. Okay, volvox, although they are not. These are also these colonies of volvox or Pandorina, whatever you take. These are forming just like this mitotic division, just undergoing mitotic division. They form multi-individual colony. I'll not call this as multicellular colony because all are individuals here in the above colony, but it is resembling the morula of our embryo. So this theory is always taken into consideration that animal have evolved from single-celled organism. Multicellular organism are evolved from single-celled organism. They are primitive organisms. And this process is repeated in our embryological development. So ontogeny repeats the phylogeny. That is the saying always, or the phrase that people always call this way, that ontogeny repeats phylogeny. What does it mean? The ontogeny is nothing but embryological development, and that indicates your pathway of evolution. So it indicates that you were earlier single-celled organism and then became slowly multicellular. And this is what the dinoflagellates show on the top. So top you find Chlamydomonas individually separate, but if I go to gonium, then they become tetrad. So this tetrad is nothing but it indicates that our evolution has come from single-celled to multicellular, and that the pathway is that nothing but we first of all colonize, and this colony became highly specialized colony, and each individual in a colony was specialized, and that's why these form a cell now instead of each cell working as individual. In multicellular organism, if I talk about comparing with amoeba, amoeba can live, grow, show irritability, they can multiply by reproduction. But single cell, if you take out from any multicellular organism, that single cell can't perform all these functions together. Okay, because it is highly differentiated cell. If I am taking blood cell out, blood cell will not function as nerve cell or muscle cell. This is because they are differentiated cells. And this makes the whole body of multicellular organism. And if any cell is separated, that cell dies because it needs the assistance of other kind of cells. But in amoeba, single cell can perform all these functions. And that's why amoeba is living organism, entirely complete living organism as the multicellular organism. So we can't compare the cell of multicellular organism with amoeba, although amoeba is single-celled animal, unicellular organism. And that's why amoeba is referred as acellular organisms. Okay, next slide, please. Now, this is the slide of coelom, multicellular organism. Sorry, I can't see. Yeah, germ layers. Okay. Okay. Now, germ layer means what? This is a case of multicellular organisms. In multicellular organism, many cells work together to give everything, serve a particular body. Just now I told you nerve cell, muscle cell, blood cells. These are different kind of cells come together and form a body of animal, and this will function in a particular manner to of survival. Now in germ layer, when embryo is developing, if you go from morula ahead, it becomes blastula. Blastula has a cavity in the center. You can see the diagram here. And blastula will proceed to gastrula. What is gastrula? Gastrula is nothing but, if I compare blastula with gastrula, blastula is single cell layered embryo. Cell layer is single. Okay. And inside there is a hollow called as blastocele. Okay. Now, if I talk about gastrula, gastrula invaginates, blastula becomes invaginated. And because of invagination, you will find there are two layers developed in gastrula. External is called as ectoderm and internal is called as endoderm. So these are the three stages in egg development, that is morula, blastula, and gastrula. Now it is fixed, I think. Now let us talk why I am displaying this all three stages here. If I talk about morula, on the right side of morula, you'll find actual 3D image of morula, how it is multicellular. On extreme right of morula, you'll find Pandorina. Pandorina is just like dinoflagellate. Just now I talked about gonium, plasmodium, and volvox. This is also dinoflagellate, and it shows exactly colony, just appearance like a morula. So it indicates it relates to morula, isn't it? So this organization germ layer is single, or it is not defined rather. In morula, germ layer is not defined. In next stage, blastula, you'll find it is single layered organism, blastula. And what is resembling example? Resembling example is nothing but the sponge. Sponge is extremely primitive multicellular organism. Okay. Sponge is nothing but extremely primitive multicellular organism. These are unable to move even. There are very feeble movement in muscle cells which are there inside. That is exclusively pore cells. There are pore cells. These sponges are referred as poriferans because they have porous body because of pore cells. Only pore cells can contract and open. Other all cells have no ability to contract and relax as such. So there is zero kind of movement in sponges. And this is single layer multicellular organism because it is primitive. So that resembles the blastula. So animal, as they develop from simple to complex form, will find they are repeating exactly our embryological conditions. Now next is talking about gastrula. It is double layered. Outer layer is known as ectoderm and inner layer is known as endoderm. And the cavity, new cavity develops because of invagination. Blastocell reduces and new cavity develops, that is called as archenteron. And this archenteron retains in higher animal. We'll talk about that in the coelom development. Here we are just concerned only about two germ layers, ectoderm and endoderm. And this type of animal directly is seen in existence in nature. Those are all coelentrates. What is coelentrate? If you talk about hydra, the figure is on your extreme right. Hydra is there. Other example usually you talk about that, that is jellyfish. These are double layered animal. They are having two cell layers. One is ectoderm and endoderm. And that's why these animal are known as diploblastic animal. Okay. Now, more advanced animal. After this, talking about phyla, if I talk about phyla, when unicellular organism or acellular organism is referred, that is protozoa. If I go to higher phylum, that is Porifera, these are single layered organisms, but multicellular, and they are referred as Porifera. Higher phylum is Coelenterata, and Coelenterata is represented by hydra and jellyfish-like organism, which are diploblastic organism. If I go to higher phylum, that is Platyhelminthes, Platyhelminthes are made up of three germ layers. Why? In addition to ectoderm and endoderm, there is a third layer between these two germ layers added. Between these two germ layers, ectoderm and endoderm. Between these two germ layer, additional third layer is added, which is known as mesoderm. Okay, that is secondarily developed. Originally, embryo develops into two germ layered structure, ectoderm and endoderm. And third germ layer, which is mesoderm, is secondarily developed with certain mechanism in gastrulation. Okay, that part will be taught to you how gastrulation takes place, and third germ layer, that is mesoderm, is developed, is be taught to you in embryology, but we are not concerned here. We just remember that third germ layer is added in Platyhelminthes, and that's why Platyhelminthes are known as triploblastic, TRI, triploblastic. What does it mean? These are in general referred as three germ layered animal. Okay, three germ layered organism. So triploblastic. So higher animal become now triploblastic. If I give you two animals which you don't know, and I'll tell you that, tell me which is belonging to Coelenterata phylum and which is belonging to Platyhelminthes phylum. So we'll just take a section, find out animal is whether three germ layered, triploblastic or diploblastic. If animal is diploblastic, you will tell that this organism is belonging to Coelenterata. And if it is triploblastic, you can easily say that this is belonging to Platyhelminthes. So it is so simple, but actually in experiment, if you go to searching such type of animal, it is a bit difficult to identify which cells are belonging to ectoderm, mesoderm, and endoderm. That is different part. But now this is simple answer I am telling you. When somebody asks you that if two organisms are there, how will you say one is belonging to Coelenterata and one is belonging to Platyhelminthes? So you can simply say that if it is diploblastic, it is belonging to Coelenterata. And if it is triploblastic, it is belonging to Platyhelminthes. And that is where the taxonomy works. Why we are referring this germ layers? Because they are showing you this particular phyla characteristic. Okay. Now on left hand side diagram, we come across one embryonic condition. Actually, it is triploblastic organism. Blue, red, and yellow color you can see. Blue color is ectodermal part, red color is mesodermal part, and yellow color is endodermal part. Now, if you read a coelom label, it will be embedded on lower as well as upper part. Either side, coelom is covered by mesodermal layer. Okay, such type of coelom is called as true coelom, that we are going to talk in the coelom formation. But just remember that. And this is triploblastic organism because it contains ectoderm, mesoderm, and endoderm. Can I request for next slide, please? Next slide. Okay. Now, tissue level organization. This is very simple. What just now I told you, that multicellular organism only can show you tissue level organization. First of all, we have seen a cellular level. Then we have seen that why animal tended to become multicellular and which are the multicellular. Then after becoming multicellular, we have seen they are made up of different layers of cells. So they may be referred as diploblastic and triploblastic organism. Okay. Now we are going to see that this multicellular organism make their cell to differentiate into different forms so that their body is formed in a complex form. For example, here upper part is that tissue level organization starts first with multicellular sponges and diploblastic cylindrids. So multicellular sponge is representing morula-like or blastocell-like structure, blastula-like structure. And these animals show different kinds of cell in their body. What are those? It may be scleroblast, it may be myoblast, it may be schizocytes and trophocytes, like the different kinds of cells are there. This trophocyte is functioning only for feeding purpose. Schizocyte is storage purpose. Scleroblast is for developing spicules in sponges. And there may be porocyte and choanocytes like that. Collar cell. Collar cell is nothing but which is functioning for digestion of the food which is taken in the spongocyte. Like that, poriferans are exhibiting different kinds of cell, and that shows that cells have differentiated into particular specialized type. Even if we talk about sponge spicules and canal system, these are nothing but differentiated cells. Okay. Now, if you talk about cylindrids like Hydra, lower diagram, there certain cells come together. For example, nerve net shown in Hydra. Okay, nerves cells come together to form a nerve net. Why this nerve net is required to Hydra and not in sponges? Because sponges can't move. They don't want to move because they are elementary, very primitive type of animal. But Hydra are little bit clever as compared to sponges. Hydra are cylindrate and cylindrate are diploblastic. And during developing, again, cell have differentiated into muscle cell and nerve cells. And muscle cells work for movement. You might have seen the movement of tentacle of Hydra. This is because of muscle cell, which is not seen in sponges. So sponges are elementary as compared to Hydra. Now Hydra also show nerve net. This nerve net help Hydra for carrying the senses of different kind. For example, movement of Hydra is possible only because of this nerve net. You know that nerve net is helping for sensory function. So that's how. And on the right hand side, again, cell wall of Hydra will show you different kind of cells. So cells have become differentiated. So tissue is formed from similar kind of cells which is differentiated. For example, nerve net. If I talk about muscle system, tissue of Hydra, then muscle tissue is developed from muscle cells. If I talk about digestive cells, digestive system of Hydra, then this kind of cell is only functioning for digestive purpose. So all cells which are specialized will unite together to form a tissue, just like hydra nerve net. Okay, next slide. Now, organ level organization. Tissue is formed, which is simplified organization. Okay, simplified like nerve net, but there is no brain or nerve cord here in the hydra. In higher animal, what is there? In addition to tissue, tissue is modified into particular specialized structure called as organ. Okay. For example, your eyes are nothing but sensory organ, which is helping for sighting anything. It is forming image of object on your brain, and this is how the eye is functioning for sense of sight. Like that, sense of smell, sense of touch, sense of taste. So many senses, sense of touch and all. These are the sensory organs. Or if I talk about reproduction, then gonads are nothing but organs. Okay, the uterus and testes, these are nothing but organs. They are specialized for particular function. So simple tissue can't do this all. They have to modify into particular organ. For example, kidney, if I take, kidney has to work for filtering your blood for waste matter. So blood is filtered in kidney, and that waste matter, that ammoniacal matter, whatever are there, and toxins which are filtered in kidney, and that is thrown away from your body. If it is not done, you will die immediately. So these organs are there. So which organs are seen in primitive organism first? That is in diploblastic organism, which is ciliated, where tissue level organization is already in existence. But in this animal, in addition, you will see rhopallium like organs, tiny organs which are developed in this animal. The above diagram is jellyfish, that is cuboid jellyfish, or cuboidal say cyphozoans, and the lower example is of a common jellyfish. In both of them, there are balancing organism because these jellyfishes move in water. When they are moving, they contract their umbrella-like structure, that is called as medusa. These are medusoid form. Okay, there are polyp and medusoid form. Hydra is polyp form. This is medusoid form. We are not talking about all forms now. Here, just we are concerned with the organ which is developed in this. What are these organs? Actually, this jellyfish and cuboidal, cyphozoans, they move from one place to another by particular muscle movement. But while movement, they have to keep their body upright. So for keeping this body upright, animal has to use balancing organ. In our body, we have balancing organ where it is present in your ear, called as cochlea. Our ear has cochlea, and that is a balancing organ. Similar balancing organ is seen in animal called as rhopallium in jellyfishes. This rhopallium has particular statolith for balancing purpose, and also certain chemoreceptor they have, and photosensitive cells are also seen in rhopallium. And because of this animal can go to any level. If it is surface water, then light intensity is high. So this rhopallium can sense light intensity is high, and because of that, this jellyfish will try to submerge inside, go inside, deep inside, because they don't want to get exposed to the surface. So they will go little deep, and they stand upright because of the otolith or statolith over there. And this statolith is nothing but balancing organ. So because of that balancing organ, this animal remain upright. So these are the tiny organ. I'm not concerned to talk about function of this, but this tiny organ is first time appearing in ciliates. Next slide, please. Now, organ system level. Just now we called organ is developed. First of all, tissue has developed. Then organ has developed. Now, organ system level organization. What is this? Many organ together form a system. Let me talk about digestive system. You know that it starts with mouth, then teeth, then tongue, then pharynx, then esophagus, stomach, then intestine divided into ileum, colon, and rectum and all. Okay. This is our one system, digestive system. If I talk about blood circulatory system, then heart, then aorta, arteries and veins, and blood itself. So this circulatory system is there. If I talk about brain in our case, then sorry, if I talk about nervous system in our case, then brain, nerve cord, and nerve nets. So many nerves are there and sensory cells on the tip. This all together function as a nervous system. Similar here in animal, you'll find system, organ system level organization in, it is first time occurred not in jellyfishes, not in hydra, because these are only showing organ level organization. But many organ when come together, it is system level organization, and organ system level organization first time appears in triploblastic platyhelminthes phylum. Okay, triploblastic platyhelminthes, where you'll come across very highly developed system, that is reproductive system. If I talk about higher phylum, that is Annelida, and representation is by earthworm. On your right hand side, the diagram is there, is earthworm, which shows digestive system, which is entirely complete digestive system, and this shows variety of organs coming together forming a digestive system. If I talk about a middle figure, which is showing a digestive as well as reproductive system, is Ascaris. Ascaris is roundworm. Now let me talk these all three examples. Left hand side is tapeworm, which is platyhelminthes belonging to phylum Platyhelminthes. Middle one is roundworm, Ascaris, which is belonging to phylum Nematoda or Ascalminthes. And next one to that is belonging to Annelida called as earthworm. So all are triploblastic, but there is a difference. First one, platyhelminthes are acylomate. Second one is pseudocoelomate, and third one is true coelomate. This thing we are going to discuss in next part. Next slide, please. Now symmetry, very simple. We are going to discuss under that basic concept and then types and then evolution. Next slide. Okay, let me tell you about the symmetry. Symmetry is nothing but if anything, any object is possible to divide into two halves, which would be mirror images of each other, is symmetry, isn't it? So on top, certain geometrical figures are there, which shows on the top two, there is an axis which divide this object into two, first of all, first figure. Second figure, there can be three axes along which this animal can be divided into two mirror images. So this is little bit complex to understand. This figure is showing you three axes, isn't it? So don't draw all three axes at a time. You have to imagine one axis at a time. Okay, one axis only. Just imagine one axis. It may be right side axis, left side axis, or horizontal axis. Okay, any one axis if you imagine, and cut that particular object into two pieces, that will be mirror image. Then forget about that axis and draw another axis and cut animal along that. You will find again two mirror images. Likewise, I want to say. So here it is having symmetry into on three axes. And the next figure, if you imagine, that figure can be cut into mirror images along four axes. Okay, four axes. So this is how you find symmetry. And the next part here, I hope you can understand the figure of tree. Some trees are symmetric in appearance and some trees are not symmetric. So trees usually have a symmetry, but some of them are typically symmetric. For example, coconut tree, they are typically symmetric. But if I talk about mango tree, it is a symmetric type of plant. Now talk about animal. On your right hand side, there is a butterfly figure. It can be divided into two mirror images along only single axis. The axis is drawn. The blue area is showing one half and the clear area on left hand side shows another half. Now talking about lower figures here, animals also show all kinds of symmetry. There are three types: asymmetry, radial symmetry, and bilateral symmetry. The butterfly is showing bilateral symmetry. Lower figures, if you refer, on left hand side, there is no symmetry, so asymmetric animal. The example is sponge. Then the next to sponge is a radial symmetry, which is represented by jellyfish. Okay. And then second last is bilaterally symmetrical is a fly or insect. And extreme right, if you find, the hydra is dividing into several planes, several axes. So here we have drawn altogether 8 axes. Okay, sorry, 6 axes here. Okay, 8 axes. All 8 axes, you will find that hydra will be divided into 2 mirror images. You can imagine any axis, all 8 axes, because this is it is having a radial symmetry, and this radial symmetry can be shown on altogether 8 axes. Okay, and because of that, this animal is having a particular, a typical radial symmetry. And now why this radial name itself is there? Radius is nothing but it is a line drawn from center to the periphery of a circle, radius. You can draw enormous number of radii on circle, but that is not possible on hydra. So because it is divided along only 8 axes, it is known as octahedral radial symmetry. Okay, next slide, please. Now talking about different kinds of symmetry, asymmetry is there in sponges and amoeba. Then radial symmetry is there in starfish. It is also known as pentamerous. Okay, radial symmetry, because starfish can be cut into 2 mirror images along altogether 5 axes. Okay, and that's why it is pentamerous radial symmetry. Now talking about bilateral symmetry, it can be shown by higher invertebrates as well as chordates. Higher invertebrates, which are those? Right from platyhelminthes onwards. Animal body becomes elongate and it is bilaterally symmetrical, because only one axis can drawn which divides this animal into 2 mirror images of part. Okay, if I talk about planaria, planaria can be drawn in middle line, longitudinal line can be drawn, and similar is the case about human being. So these are bilaterally symmetrical. On either side, they are identical or mirror image of, along either side of the axis, you will find mirror images of the body part. Now, lower side, if you see the internal part, which show the bilateral symmetry. Bilateral symmetry can be external as well as internal. So human body internally shows bilateral symmetry by having pairs of arteries, pairs of lung, pair of kidney, pair of brain part also. Limbs are also paired. Like that, bilateral symmetry is seen in human. Like in lower organism like planaria, if you see nervous system is dividing into two longitudinal nerves which give horizontal nerves, which show symmetry. Next slide, please. Now we're talking about symmetry, evolution of symmetry. As we see that sponges and protozoans show no symmetry, so they are asymmetric organism. And as we go higher level, we go on. See, first of all, we see radial symmetry and then bilateral symmetry. So this diagram on left side, it's indicates that sponges are very elementary and they are asymmetric. Cnidarians radially symmetrical. From flatworm onwards, flatworm, pseudocoelom, and coelomates. These all are bilaterally symmetrical. So bilateral symmetry relates to highly developed organism as compared to radial symmetry. And asymmetry is primitive to radial symmetry. Next slide. Evolutionary significance of symmetry is that uh, compared to asymmetry and radial symmetry successful, these animals are radial symmetry is more successful as compared to asymmetry. And if I talk about bilateral symmetry, bilaterally symmetrical organisms are more successful as compared to radial symmetry animal. So bilateral symmetry seems to be part of the embryo forming as one segment is affect the previous and original segment. This repeat iteration of the previous or original segment. This segmentation allows for a basic plan that is then modified in particular segment and specialized, just like cephalization. So in bilateral symmetry, animal gets one benefit that it gives rise to segmentation. Okay. And because of segmentation, they maintain their development of appendages, and also head development is possible because bilateral symmetry defines anterior and posterior side, lateral side, as well as dorsal and ventral side. It is not seen in radially symmetrical organism. Radially symmetrical organism has only two sides, that is ventral and dorsal. Whereas bilaterally symmetrical organism has anterior, posterior, two lateral sides, left and right, as well as dorsal and ventral. Because of this, all sides develop. Animal has chance to develop particular organ towards anterior side called as head, and this is helpful for animal for movement, segmentation, and head development that we are going to see later. So first of all, symmetry, how it is beneficial to in bilateral symmetry organism. Cephalization is also possible for highly moving organism. For example, cheetah, if you see, head is highly defined in this organism, and because of head highly developed, the cephalization is advanced, and because of this, animal can move faster. Limbs also helps in bilateral symmetry. Next slide. Now, coelom, basic concept, then formation of coelom and type of coelom. Next. Next slide. Now, here, if I talk about sponges, they look like blastocyst. This is very primitive type of coelomic cavity. Actually, it is not coelomic cavity. It is body cavity of sponge called as spongocoel. Okay. Next is the gastro-like organism bilayered called as diploblastic organism called as diploblastic coelentrates. In coelentrates, the archenteron which is developed in the gastrulation, in primitive gastrulation, is retained as it is. And it works as digestive as well as coelomic cavity of coelenterate. That's why it is referred as coelenteron. Coelenteron. Now, coelenteron meaning coelomic cavity, coelen, and enteron refers to digestive system. Coelen refers to coelomic cavity and enteron refers to digestive system. So digestive system as well as body cavity, coelom, both functions are done by this archenteron. That's why it is known as coelenteron, which is not a true coelom. Spongiocele is not a true coelom, which is coelom here on the next part. You will see the coelomic cavity in human being, for example. The extreme right part, if you see, is developed into internal body cavity, which is called as coelom. Okay. Next part. Next slide. Now, in this embryonic condition, coelom is developed in particularly triploblastic organisms. No unilayered organism can develop coelom. No bilayered diploblastic organism also can't develop any coelom. It is triploblastic organism, that is 3 germ layered organisms, they can develop coelom. Now, how they develop? There are 2 methods. One is schizocele and other is enterocele. Enterocele, on right side diagram, if you find left hand side diagrams, that shows that gastrula develops bilayered embryo, diploblastic embryo, and while developing mesoderm, the coelom starts developing itself. Okay, and that is shown on your right side diagram. In right side diagram, you will find that upper left hand side process, left hand side, if you see upper and lower, is titled as protostomes, and right hand side series, upper and lower diagram, that is titled as deuterostomes. Okay, upper title you refer, you will find protostome and deuterostome. Just observe protostome. Protostome will show its coelom development from mesoderm, but it is by splitting of mesoderm. Mesoderm splits into 2 layers, that is lower layer and upper layer. Upper layer will lie close to ectoderm and lower layer will lie close to endoderm. And this splitting up, inside there is a cavity which is developed, is called as schizocoelom. And the process is known as schizocele. And it is commonly known as the coelom formation by splitting of mesoderm. Schizocele is nothing but where the coelom forms by splitting up of middle layer called as mesoderm. So mesoderm splits up into outer layer and inner layer. Outer layer lies close to ectoderm and inner layer lies close to endoderm. And this forms the coelom called as schizocoelom. In deuterostomes, what is the condition? There is no splitting of mesoderm. Here what is happening? There is a sprouting up or budding up. A blind tubes are or blind sacs are developed at the end of gut. Usually gut is made up of endoderm. And endoderm gives rise to certain outgrowths, finger-like structure outgrowth. These outgrowths later on develop into a coelom. They get separated from endoderm and form a separate inner layer called as mesoderm, red part. And this mesoderm leads to a coelom formation. As it is formed from the gut, this is called as enterocoelom because enteron refers to gut. Okay, digestive system is referred as enteron, coelenteron, remember, coelenteron. So enteron is referred to digestive system. So enterocele develops in deuterostome by separating from digestive system. Whereas in lower organism protostomes, mesoderm develops separately by differentiation, and that mesodermal layer splits into upper and lower layer, and this forms the coelomic cavity called as schizocili. I hope that you have understood the difference between schizocili and enterocili. Next slide, please. Now, in true coelomates, okay, this is also repetition of protostomes and deuterostomes. Let us talk about this. If I am zooming, can you see? I have zoomed the figure. Is it seen zooming? If I am zooming that, no. If I zoom it, whether you can see I have zoomed up.
No, sir.
I just want, no. That is the problem of presentation, isn't it? If I had presented same slide, I would have shown you what
Yes, sir.
exactly I wanted to show. So this
Yes,
is not possible if somebody other is operating. Sorry for this. Now I'll have to go with my explanation only. Left hand side figure shows a deuterostome and protostomes. Protostomes showing the schizocili and deuterostome shows enterocili. But I want to tell you one more thing about these animals. Protostomes are named to the animal who develop their mouth from blastopore. Okay. When blastocele is formed, when gastrulation is formed, gastrulation develops invagination because of invagination, and it forms a blastopore. Okay. And that blastopore, if it develops into mouth of organism or animal after development, then that organism is known as protostome. Whereas in deuterostome, blastopore develops into anus, and opposite to anus, there develops a separate opening called as mouth. And hence, these are known as deutero. Deutero means next, duplication, or second formation. Because of that, deuterostomes are highly developed organism as compared to protostome. And this deuterostome and protostome are often used in taxonomy. Next figure. Next slide, please. Okay. Now, types of coelom. Animals are divided on the basis of presence of coelom. If it is present, then they are known as coelomate. If it is absent, they are known as acoelomate. The first example on left-hand side is acoelomate, that is flatworms. All flatworms are not having any coelomic cavity. Rather, their coelomic cavity is filled up with parenchyma cells. And because of that, these animals are referred as acoelomate. Second figure would refer to pseudocoelomate organism. Why? Because this coelom in this animal, these are the example of roundworms, ascaris, for example. So ascaris do not show true coelom because coelom is not formed by mesodermal on either side. If you see true coelom on right hand side figure, true coelom is lined externally and internally by mesodermal layer. Okay, pink color mesodermal layer on the right hand side figure if you see. But the case is middle, that is that pink layer lies on one side towards ectoderm, and endoderm is not at all covered by mesoderm. Because of that, this particular coelomic cavity is known as pseudo or false coelomic cavity. Pseudo का मतलब क्या होता है? It is false. Why false? Because it is not true. What is true? True is the coelom which is having mesodermal layer on either side of coelom. That is body cavity. Body cavity, if it is covered by mesodermal layer on either side, external and internal, then only that particular coelomic cavity is known as true coelom. But in pseudocoelomic animal, mesoderm lies on one side of coelom and on other side endoderm is present. Because of this, the pseudocoelomic animals are different as compared to true coelomate organism. What is the example of true coelom organism? These are annelid onwards. Now, why this is studied here? If I am studying taxonomy, if I tell you to differentiate between Platyhelminthes phylum from Annelida, so you will be able to tell that annelids are triploblastic, but even Platyhelminthes are also triploblastic. But what is main difference between them are there? Annelida has true coelom, whereas Platyhelminthes has no coelom. So you can call Platyhelminthes is a coelomate and Annelida are true coelomate. These are the major difference between these two. Again, if I talk about coelomate, Ascalminthes, Nematoda, and Platyhelminthes, then the difference is that Platyhelminthes has no coelom, so it is a coelomate, and Nematoda or roundworm show coelom, but it is not a true kind of coelom. Okay, that's why these are known as pseudocoelomate. So Ascalminthes are pseudocoelomate and Platyhelminthes are acoelomate. Next slide, please. Evolutionary significance of coelom is varied. First of all, in earthworm, if we study our function of coelom gives turgidity to body because coelom always is filled with gas or fluid. And if it is filled with fluid, fluid gives particular turgidity to body, and this may function as an internal feeble skeleton of the body. And this gives a particular shape to body, isn't it? That is one. Second thing, that coelom provides protection to internal organ. That was the main cause why coelomic cavity has developed. Internal organ, if they are small in embryonic condition, it gives a particular space for developing this organism that will grow and lodge to particular area. Okay, liver, kidney, lungs, heart, these are lodged into particular cavity, coelomic cavity. That is how significance is. Third significance, evolutionary significance, is that it helps in locomotion. If you try to see earthworm movement, there are certain videos available in on YouTube. You will just search earthworm movement. In that video, you will find that earthworm can move by contracting its body and bulgifying, but swelling and contracting, swelling and contracting. This is the way how this animal moves from posterior to anterior. And animal can move by just extending its body by contraction of all the segments. It extends body, then anchors to the substratum and pulls posterior body by swelling. So this movement is very interesting. And that movement is possible only because of coelomic fluid, which is pumped from one segment to another segment of earthworm. Okay. And this is very nice, fine mechanism how it is performing the movement of earthworm. So that is also one of the kind. And not only that, earthworm is very miserable and feeble animal. But if you talk about higher animal like insects, they can fly. Okay. The coelomic cavity which is developed inside supports the lateral part. Okay. So it supports segmentation first of all, because segmentation is possible only because there is coelomic cavity present inside. If coelomic cavity is present inside, earthworm, like first of all segmentation takes place, and that evolves into bilateral symmetry, and bilateral symmetry later on gives rise to development of appendages just like wings or legs, and these appendages can make animal move faster. So locomotion is associated with coelom formation, isn't it? So these are all together very important for development of coelom, and that must be the reason why coelom has developed inside the animal, higher animal. Presence or absence of coelom and the way in which it was formed was believed to be important in understanding the phylogenetic relationship of animal phyla. However, recent molecular phylogenies have suggested these characteristics not as informative as previously believed. So, although coelom formation is showing your phylogenetic path, for example, whether animal is a coelomate, pseudocoelomate, or true coelomate. These are the three stages of coelom development. Earlier, taxonomists had taken into consideration that this coelom development from acoelometry to true coelometry, this process have given rise to evolution to animal. Evolution animal has come from
एक्सक्यूज मी
acoelomate to
सर. हॅलो
yes.
सर, आपला 2चा दुसरा एक लेक्चर आहे 2 वाजताचा, आणि आपल्याला
Okay. No problem.
थांबायला हवंय सर.
Okay, 2 minutes. And sorry for this. We have already lost our 15 minutes. So I can't help. 15 minutes was enough for me to finish our lecture. Anyway, thank you very much, all the students. If you can have an access to my PPT, which is sent to organizers, you can get the PPT and further study that PPT for detailed levels of organization. Thank you very much for giving this opportunity to the Dombivli College and Dalvi College. And I'm very happy to share this knowledge with you today.
थँक्यू सर.
Thank you.